research is questioning and disagreeing. I'm Mayla Boguslav and I study research. I ask questions such as, what is research? How do we measure its success? I live by the motto, ignorance is bliss, because when you don't know something, it's an opportunity to learn. Researchers live in the ignorance or the unknowns. They ask questions, identify areas of ignorance, lead the way to gain knowledge, and most importantly, ask the next questions about our new ignorances. Welcome to the Ignorance Podcast, where we learn about research through research questions. Let's welcome our guest researcher for today. Please introduce yourself by name only and your favorite thing to do outside of work.
My name is Julie Horvath, and outside of work, I love to do many things, but most of them involve nature. So I love to travel and explore the world. Hiking is a big pastime.
Do you have a favorite hike or do you usually do different ones?
I try to do different hikes, but I do have a state park near my house, and there's one particular hike that my family and I like to do because it gets us near water and goes through the forest as well. So great place close by home.
Yeah, I love nature as well. It definitely is a nice reset on the brain for anything and everything, which is really nice. So what questions excite you in your work? What questions are you currently pursuing?
So great question. Many things excite me, which is a challenge because then I try to do too many things. So I've always been really passionate and interested in genetics and the connection to disease and connection to behavior. And so some of my more recent questions have actually addressed looking at the genetics of different species and how that impacts their health and behavior, but also how the microbes that live on our bodies impact our health and behavior. So that's where I'm going in the recent past, was starting to look at the microbes on humans and other animals.
What has motivated thinking about that question?
I guess just a curiosity, fascination. I mean, I love mysteries, and I until recently didn't even really think about the microbes that lived on and inside me. And so now thinking about, oh, there are all these tiny little microorganisms that live on us. What do they do for us? You know, we used to think of them as kind of negative and bad and causing disease, and we're now starting to realize that a lot of them are beneficial and we need a lot of them to survive. So got me really curious about which ones are they? What are they doing? What do we know about them? And there's a lot that we don't know yet. So I think that also fascinates me because I always want to be the first one to find something new. It's probably what motivates me as a scientist is, trying to figure out, well, what does nobody know? And I want to go find that answer. And I want to be the only one that knows it. And then realize, well, probably there are only 3 or 4 other people who care that much about it. But I get really passionate about it because then I'm, an expert in whatever topic I'm exploring.
Do you have a favorite microbe right now?
Goodness, I don't think I have a favorite microbe. One I study a lot are different types of Staphylococcus. So Staphylococcus is one that lives on the skin. That's a genus level. There are multiple species in the Staphylococcus genus. So there isn't one particular one that I love to study, but I'm now going to think about that. Who's my favorite microorganism?
Yeah, it's really interesting, these like small little bugs that live on us. And we have probiotics and antibiotics as one example I think of a lot, where antibiotics is like anti the bacteria and probiotics is pro the bacteria. And we really need both, yogurt being a great example of probiotics. I've definitely taken antibiotics and needed to take probiotics afterward because it just ruins my gut system in relation to digestion. So very, very important. With this research or in general, how do you hope to impact the world?
So that's a great question. As a scientist and someone who does a lot of teaching, I taught at the college level for quite a while now. And so often training students and figuring out how do you teach them what the important information is, how to teach them to think about some of these questions, get them thinking about how they can impact the world as well. And so that's kind of how I feel I could fit in one role. But until recently, I also worked at a museum of natural sciences and just thinking a different way. When I was a college professor, I was just in the academic world. And then in this museum, now I started engaging the public a lot more, starting to think about how do I get them to appreciate science, understand what the scientific process is, and really appreciate, hey, this is why we should fund research, for example. This is why it's really important, and also getting them to really understand it. So when I was in the museum, I worked in this glass-walled lab. And it was really interesting because as you're inside the lab, you don't really think about people watching you. But since it's a glass-walled lab in a museum, the public can come up to the window and can watch you do things like spill pipette tips all over the floor, for example, or do something really silly that you didn't realize somebody, or 10 people saw you do that. But it's a really great opportunity to think about engaging the public in what your science is. But then it also makes you really think about what the value of your science is. Like, what's the value of your research? Why should anybody care? Why should we even get funding to do this research? Because sometimes it might seem silly or inconsequential, but there are often larger questions that you're trying to address when you ask a really specific question. And so just trying to convey that to the public was really insightful, and also really got me thinking about my value as a scientist in the world.
Um, Kind of last year, like my postdoc at Colorado State University, I learned more about museums through an education lens. And it's a really cool space to think about how we communicate science to the public and how we teach science to the public, not just in schools, but also adults, continuing education for anyone who comes in, and also the multi-generational piece. So I guess, can you speak a little bit more about museums and what does it mean to have a lab in the middle of a museum? How also did you showcase the question that you're asking, right, to motivate what you're saying exactly?
So it was set up as a space where I was doing my research just like I was in an academic institution. So we had a lot of standard molecular biology equipment and microbiology equipment, lots of pieces of equipment that you would see in an academic space, but it was laid out in this museum setting so the public could kind of see the instruments. And so we had to really think about, how do we portray this? And so we would put signs up with explanations about some of the equipment. This is a DNA sequencer. This is what it does. This is why we're using it. These are some of the questions we can ask. And here's how we funded it. We had to write a grant to the National Science Foundation to get funding for this. And now we use it for our research, but we also use it to train students and train the public who come into the museum to maybe take some classes or learn something about our research. So it was really interesting to try and think about how you connect to the public. We did have a lot more things in the museum, a lot more pieces of paper explaining things, a lot more posters where we'd explain what we were doing, or examples of some of our experiment. If we were looking at the microorganisms that lived in cheese, maybe we could have samples of cheese, or creating kimchi or kombucha. And so we'd have jars of material that was, an example like, hey, this is actually a kombucha. This is fermented tea, and this is what we're doing, and this is why we're asking some of the questions that you see us working on behind the scenes. And so it was a really interesting way to connect with the public, and then just trying to find additional ways to excite them and also explain what the science was, because I think there's a huge disconnect between a scientist doing an experiment and then the public thinking about you publishing it. And so I think a lot of times the public thinks, oh, you do one experiment and then you write it up and then submit it to a journal and it's published, and don't really think about, like, sometimes it takes months to do different experiments, come at them from different angles, so that if two somewhat different experiments come to the same conclusion, then you have, more confidence in what that answer is. And so sometimes you're thinking about different ways to do experiments and really showcase how you do that and what the scientific process is. I know that was a long-winded answer.
Oh, that was great. I know museums for me definitely sparked some curiosities for sure, and especially changing museum exhibits. So yours is kind of a changing exhibit as you're doing science, which is pretty cool. And it's interesting to think about when somebody comes in, they could be at any part in the process that you're doing. I definitely have seen live labs. I've more seen it in relation to archaeology things where they're like cleaning bones or other things like artifacts. But it's really cool that you're also doing this and showcasing this to the public, and just the different things you have to think about in relation to that, that I also wish in general more scientists thought about at large, and that we were more communicating some of these things to the public. This is kind of part of my goal of this podcast, is to be able to showcase really a lot of research is about questions, not always about answers. Yeah, so clearly you've influenced a lot of people in relation to sitting in the lab, right? Maybe you've thought about quantifying that if you want to share that. But I guess, who has influenced you? Who are people you look up to? Because it's all a continuum of people before us and people after us. And I'm sure there's many people you've excited to do science and work in different spaces, just sitting in the museum, and other research that you've done. But I'm curious how you got there from your process.
I mean, many people have influenced me, and Some of the influences have changed over the years, obviously. When I was young, I was really passionate about animals. I wanted to help animals and potentially be a veterinarian. Then when I realized, okay, that's probably not the right profession for me, but I still wanted to do something to help animals. And so people like Jack Hanna, who are really captivating and would go on, shows like The Tonight Show with all these exotic animals and really captivate the public. This is why these animals are important. They're, maybe in danger of extinction. And here's why we need to actually save their habitat and do other things to really promote them. So people like that really got me thinking about how do you captivate people? It's going to help you a lot more in talking about your science if it's new and exciting and also relevant to their daily lives. So if you talk about something really abstract, people kind of glaze over and don't want to hear it. Thinking about people like Jack Hanna who made, any animal really exciting and captivating. And so how do you do that with science? People like that who are great science communicators are people I definitely have looked up to. But one of my first mentors was Karen Frederici, who was at Michigan State University, where I did my undergraduate. And she was really amazing because she had 2 labs across campus. And so she would manage her time between these 2 labs and kind of just seeing that dichotomy of, okay, she has 2 different lab spaces and she has multiple different projects in those spaces. How does she manage and do all that? And I've kind of always modeled my future career around that. I didn't only want to do one thing. I have many different interests. And so I get really excited about all these different things and get my hands in all these different pots and kind of thinking back to her, how did she manage these 2 different lab spaces? When I was in undergrad and then grad school, I had 2 different angles to a lot of my work. And now I've continued that. So I do a lot of primate genetics work. And now I'm looking at skin microbiome research. And so those are somewhat different. I've kind of found a way to marry them and merge them. But Dr. Frederici really helped me come to terms with this is possible. You don't have to just focus on one thing. You can actually do multiple different topics and find threads where they fit together. So that has been something that I've just stayed passionate about and always had a dual job where I'm doing multiple different topics or have multiple different roles to keep me entertained.
I relate to that. I also don't like to just do one thing, and lots of questions excite me. I'm now curious, actually, what is the connection between primate genetics and skin microbiome?
There are many ways you could think to connect it, but I've started looking at primate genetics across many years. That was a lot of the work that I had done in my graduate work, looking at how different primates were related, look at their taxonomy, what are their evolutionary relationships by looking at their DNA sequence. So primates that have very similar DNA sequence probably are more related than ones with distant sequences. And so that was one of the threads that I started looking at, and then started thinking and bringing in that behavioral piece. Well, how do the genes or gene variants of some primates actually impact their disease and their behavior? And humans are primates, so humans are part of this mix. And so not only thinking about which genes you possess, but also thinking about the genes that are on the microbes that live on us. How do those interact? I've started looking at some of my research questions to address that. Like, how do host genes of me or a primate actually impact the microbes that live on me or that primate? And then once our genes maybe and our physiology impacts which microbes live on us, then how do those microbes again impact us? So how do they impact our health or disease? And then maybe how do they impact our behavior? Some of my research questions are really getting at that piece, like looking at which genes and gene variants are in different primates and how that impacts the microbes on their skin potentially. And so those are some of the newer, more interesting questions I've been looking at research-wise recently.
That's really fascinating. I know this is a podcast about questions, but now I'm curious, do you have any answers to these questions yet? And as a follow-up, how do you know if you've been successful at your work as a researcher? What is your evaluation criteria to know that you've gotten to an answer?
I'll answer that one first, and then we can circle back to any other ones I found. It's interesting to think about how are you successful? How do you know if you're successful? Certainly as an academic and a scientist and thinking through just trying to explain my research, get my research published, get my research funded, so writing grants. So I'm successful if I can obtain some research funding to do my research, support students, and then also publish or write up my results so that it gets out into the world. But the other pieces after working at this museum, thinking about how can I make it relevant to everybody's daily life? Like, how is it relevant to the general public who are funding some of this federally funded research? Those are some other pieces that I now think about, like, how does that make me think about my success? And so when we are at the museum thinking through, okay, if I sit at a table in the museum, maybe on one evening where we have a special event, how many people do I talk to and how many of them seem engaged? How many ask me questions? Because that means that they're engaged, right? If people just come up and talk to me and then leave, they probably weren't that excited. But if they start asking questions and they stay longer and then they ask more questions, then that's a really good indication that you engage them. Some of the research questions I have now have actually stemmed from talking to some of the public in the museum. They give me some anecdotal evidence that gets me thinking like, oh, maybe that would be a really interesting question to research. Why don't I, change some of my research direction or focus a little bit more effort on that and see if, other people are also going to help me support some of those research questions. That has been a different change in my criteria for success, is thinking about engaging the public and really getting a broader audience interested in what I'm working on.
So what have you found?
So many cool things. Some of the skin microbiome work that we've done has been really captivating and interesting and also engages the general public in multiple ways. I was teaching a course, this was about 8 or 10 years ago, and one of my colleagues connected me to another colleague who was also teaching a course where we wanted to really engage students in being part of the research process. And so we started this project looking at which microbes live on the skin and connecting that to one gene variant that people have. So there's one gene that controls the type of earwax you have. Did you know there's different types of earwax?
I did not know that.
Okay, there are at least 2 types, which is super interesting. I think there are probably more, but there are 2 recognized types. There's dry earwax and wet earwax. Dry earwax is generally white or gray and kind of dry and flaky. Wet earwax looks a little more sticky and is often yellow or kind of brownish and is really like a sticky or wet consistency. And it turns out there's one gene variant, so there's one single base pair change in your genome that dictates whether you have the wet or the dry variant. Okay. It's really interesting because this wet or dry variant we started looking at, we actually asked some of the students in our class, can you help us collect some samples? We collected samples from the general public and we got samples from the skin. We actually looked in the armpit as well as the ear because you have these specialized glands called apocrine glands or specialized sweat glands. And they are concentrated in your armpits and your ears and several other places on your body. And this gene that I'm talking about is expressed in these apocrine glands. So if we collected microbe samples from those places, then probably the gene product is there and it's impacting which microbes live on you, impacting which ones live in your armpits and your ears. So that was a really captivating way to engage the public because you talk to the public about earwax and people are like, oh, that's so gross. What are you talking about? But then tell me more. It's like that train wreck that they can't stop asking questions about. It was a really great way to engage students as well because if you say, hey, what kind of earwax you have, then of course they all are poking at their ears trying to figure it out. But then we kind of say, hey, there are different microbes that live on you if you have wet earwax versus if you have dry earwax. And wouldn't you like to swab your teacher and find out what kind of earwax your teacher has and see what's growing on your teacher? And so then we can actually swab the teacher and then grow it on a petri dish and see which microbes are actually growing on their skin. So the students are really captivated and engaged. We did find that people who have wet earwax versus dry earwax actually have different microbes living on them to some extent. So on your skin, you have a lot of staphylococcus types of organisms. So like Staphylococcus epidermidis, Staphylococcus hemolyticus. Sometimes you have Staphylococcus aureus, which is one that often causes disease. That's one people have heard about. But you have different types of microbes depending on which type of earwax you have or which variant you have of this gene. That was a really interesting finding that we are working on continuing to publish on and continuing to study, because it's really interesting to think about, like, one gene variant that I have is going to impact which microbes live on me. And then I just said it's going to impact which kinds of microbes live on me that also may be associated with certain diseases. So maybe they're associated with skin diseases, and some of them are also associated with ear infections. So now that's setting up some of the genes that I have that affect my overall physiology on these types of sweat glands that are producing sweat in specific parts of my body are maybe also impacting some of the microbes that live there and that are making me more or less susceptible to certain diseases. And so that piece was really interesting, that there's this direct connection, which we were assuming. But the interesting piece was we started asking people questions. We have a lot of citizen scientists or public participants who engage and provide some samples to us. And we would ask them a series of questions. And one of the questions we asked was, do you feel like you get bitten more or less by mosquitoes than the average person? And it turned out that there were some people who thought they got bitten by mosquitoes more than the average person. And they also had dry earwax. That was really exciting because it was now thinking about, if you get bitten by mosquitoes more than the average person, you're probably more susceptible to some diseases that mosquitoes carry, like malaria, dengue, Zika. And so now, if the microbes that live on your skin are impacting your directly impacting your disease susceptibility, but also impacting if you get bitten by mosquitoes more or less, then it's also indirectly impacting whether you're more or less susceptible to some of these diseases that mosquitoes carry. That direct and indirect connection was really exciting to us. So that's one of the angles we're following up on now, thinking about how can we actually make this more valuable to really help us understand how we can mitigate some of these diseases and really potentially change our impact or change some of our disease susceptibility.
Who knew earwax and mosquitoes were so related? I'm definitely thinking about, like, which earwax do I have, and like, am I more susceptible to mosquito bites? So fascinating. Like, I would not have guessed any of those things that you just said. And that's actually one of my favorite questions too, of what has been your most surprising result? Because you literally just said that you're continuing to follow these up because they're really fascinating. Can you speak to why you asked the question about mosquitoes? Like, what was your logic there? Because that is not where I went when you talked about earwax to mosquitoes. Can you fill us in a little bit more?
Totally fair. So one thing I should have said is that the microbes that live on your skin also produce your body odor. And so your sweat doesn't have body odor, but the microbes that drink your sweat, eat your sweat, then they'll give off different byproducts that produce your body odor. And mosquitoes in a big group setting, they'll cue into heat, carbon dioxide exposure, and other factors when they're trying to find a host. But one of the things they cue into when they get closer to you is the way that you smell. So what your body odor is. The microbes that live on your skin are making your body odor, and that's also a signal to certain mosquitoes. So that was one of the things we started thinking about, like, what other aspects do we have to think about odor? Several years ago, there were some researchers that actually looked at mosquito preference for different people in terms of preference for which microbes they might like, some of these volatile chemicals that get exposed from some microbes. So that was one of the questions that kind of got us thinking. There were a series of maybe 60 questions we asked, and some we thought would be really a strong connection, and then we see nothing with some of the microbes that live on skin and the answers to people's questions. So again, some of them were maybe more surprising, but the one, the mosquito connection was surprising. That's one thing that we want to follow up on and do some more testing with that, because right now we just have this anecdotal report. I mean, you also have to tease apart that maybe they aren't being bitten by mosquitoes more. Maybe they just perceive that they're bitten more. Maybe they swell up from a mosquito bite more. And so that makes them more aware of getting bitten. There are some nuances to that we also want to explore. But there's some correlation there that definitely warrants further research.
I'm really curious now to know, because then I'll figure out what my susceptibility is to getting bitten by mosquitoes in relation to that, which I would definitely appreciate sometimes to know when to put on bug spray or not. Yeah, so fascinating. It's definitely not where I thought you were going to go at all. So I love how research can go in so many different directions. especially in totally unexpected ways that really also pique our interest. And to your point about engaging communities, right, I think those are also the pieces that make some people really excited, right? When you're like, oh, earwax and mosquitoes. Did you know they actually go together, right? And people want to learn more. And so I really like this idea of kind of using surprising results in this way, or kind of things that seem a little gross to get people excited by things. Because I actually, even the name of this podcast is the Ignorance Podcast, and it's intentional because we think of the word ignorance as negative, but it's not actually defined that way. And so it makes people turn their heads and want to look more at what's going on. So I also use that technique myself. The other side of the coin of surprises is, what is your favorite disagreement?
So that one's tough because I don't have a favorite disagreement, but certainly recognize that this is one thing that drives research forward and science forward, right? If you support findings that have already been found, people aren't as aware of them or don't pay as much attention. But if you refute something and say, oh, you're wrong, then that stirs the pot, right? And so then everybody gets excited and interested. And so some of those disagreements, I think, are what moves science forward. And so recently, now it's been several years, I think, there were some reports that came out saying that there was a placental microbiome. So the placenta was not sterile. There were microbes associated with the placenta. And some people were like, oh my gosh, this is crazy. How could you ever think this? And then, scientists said, oh, well, our techniques for looking at which microbes live where, like they're getting better. So we are now finding this connection. And within a couple years, there were a lot more people doing more research in that area and started saying, hey, this is probably just contamination. And some of our techniques are so sensitive, they're now picking up some contaminants. And so this was a really interesting topic for me because, as I said, I was more trained in primate genetics and human genetics. I wasn't as aware of the microbiome until I started working in the museum and got into this space. So just thinking about the microbiome and this controversy happening as I was learning the microbiome topics was really interesting to me because I really saw it fuel a lot more research, a lot more controversy and discussion about the microbiome. It got the public also engaged more in the microbiome. I think that was one thing that really opened my eyes a little bit more about if you really want to engage people, come up with something that's novel and different and maybe is not in agreement with something that other people have shown, because that's where you're going to get some more traction and interest. Some of it is going to be kind of negative interest, right? There were definitely some challenging discussions around that, but it was also really interesting to get more people interested in this topic and got some of the really prominent names in the microbiome field thinking about, well, how does this impact our thoughts of immunology and our thoughts of creating sterile animals? And it really opened up a lot more thought process. And so I think that was one thing that I was really interested in.
Yeah, and just to say, the placenta is the sac of fluid around a baby when it's in the womb. Wow, like I've never thought about that. So have we figured out an answer to that question of, is there a microbiome in the placenta?
It's still an ongoing debate because I think some people still say, hey, we are finding this in our research. We're finding that there are microbes when we collect some of these samples, and then a lot more people are saying, no, it's just contamination. I think this is an interesting disagreement that maybe more people are leaning one direction, but it's still on.
Stay tuned for the results. So if you could do anything not limited by grants or other factors in your space, what would you be doing? What is your vision for the world?
Great question. Not limited by anything. I'm really passionate about not just doing my work, but also really compiling some of my research with others and really getting the team of collaborators, and I mean that like in a very broad sense, thinking, there are all these different scientists doing different pieces of research that fit in a really sometimes small silo. Is there a way to bring us all together and try and pull our results, pull that together so we can actually pull pieces of information from everybody's research to really answer really large questions? Is there a way to do that? I mean, not limited by money. It would take a lot. It would take a lot of coordination, right? Because no one person would be able to compile all this. No one person would be able to database and catalog all of this. But I think that's where some of these pieces of research would really benefit, because then we could actually pull together some of these pieces of information and really ask some really broad questions. And there's some really great thinkers out there who do actually pull data from other people, maybe don't only generate their own, but kind of try and make this meta-analysis. And so thinking through, like, how do we actually do that on a larger scale? So maybe reuse some data from this person, this person, this person, and then bring it together in this larger space. And I think I'm saying this because I have a couple projects where this is kind of what we're thinking about doing. How do we do this? How do we bring a community together who studies a topic and get them to work together to generate, a gigantic database, a repository where we have all this information so that other people can now start using it. As obviously, there are a lot of challenges to that. One of them is that a lot of people feel like I collect these data belong to me, and I don't want to just share it with everyone. But that's one thing that we have to think about how we're going to get around that, because once you publish, you want to share your data, and now you make it open and available for everyone. And is there a way to kind of capitalize on that and make it more useful? So you found whatever your results are really exciting, but can we actually make that more valuable now because other people can find a different way of looking at it? That would be my vision, is like getting more people to work together. And I know you and I have talked a little bit about team science. And so just thinking the team science realm, can we bring together researchers from all these different fields that would bring in really disparate data sets to answer some big questions? I think that would be really exciting.
I was going to say that this is how we know each other, is through this collaboration space or team science space. I love that vision for the world. A lot of what I hope that I'm doing with my research in this team science space is, in a world where it feels like a lot of things are really polarized right now, we're trying to actually bring people together and talk. And so that makes me feel like I'm doing something to contribute in that way. And so I completely agree. One other project I would really love to do is kind of map all the different disciplines and what types of questions they ask, what approaches they have, to understand, like, what is the best way for these people and disciplines to really fit together. Something I've heard that I would be curious your thoughts on is, disciplines really exist for academics to be able to give degrees, to be able to give a major. But at the end of the day, everyone works on so many different projects. Once you kind of come out of whatever discipline you studied, you may sit in a specific one, and I'll let you introduce yourself soon with all your titles and whatever else you want to share. But that doesn't limit us, right? Ideally, we tend to work in so many different spaces. And with this podcast, I say to people, try to guess the field. And most people say they can't because it's not obvious. And there's many different fields cross-talking with each other, especially now in this big, huge data situation that we're in, right? Where not even one person can really read everything anyways, even within one discipline. So I guess I'm curious your thoughts on like disciplines at large in relation to this idea of collaboration, how maybe you go about that piece.
It is a challenging question because I think you're right. We create these disciplines to fit in a box so that we kind of know what goes under the genetics or the biology or the chemistry, et cetera, or the history or art or whatever. I think there's a lot more blurring in those lines. And I think the team science piece really tries to bring together these researchers who work in these different disciplines, but a lot of times people are straddling multiple disciplines. And that's the exciting piece, is that once you're trained in one, it gets you thinking about some of these others, and finding connections to them is really valuable, and making some crosstalk is really valuable. So there isn't a great answer from me in terms of that, but I think we're starting to realize that we need to kind of blur some of these lines, because a lot of people fit into more than one of these, boxes per se.
Yeah, and something my last guest said that I really love is, I used to say, I don't want a box. I don't want to sit inside a box, because we say think outside the box. But David Brandman from the last episode said, actually, you want to collect all the boxes. And I like that, because then you collect all these things, you learn all these things, and you can do many new cool things with it. Every time we build something, whether it's boxes or Legos or whatever type of building block you have, you create a new structure every time. So I love your idea of this vision. And I think you and I are trying to do that in this team science space. And I hope it continues to come to fruition in all of these ways. With that, I'll ask you to introduce yourself with your name, your job title and field, and anything else you'd like to share.
So my name's Dr. Julie Horvath. Job title, I'm a senior research scientist. I work at the University of North Carolina at Chapel Hill in the Renaissance Computing Institute. I was trained as a human geneticist but I did a lot of comparative genomics, and now I'm getting into the microbiome field. So again, my boxes have maybe shifted, but I do like the idea of collecting boxes because I'm very engaged in behavior. I started studying behavior, though I am not a behavioral scientist, and realized quickly that that was not the field that I wanted to get into. I want to collaborate with people who do that, but I was really excited about the genetics piece and the microbiology piece. As you mentioned, I'm involved in team science. I'm involved in the Triangle Center for Evolutionary Medicine and UNC Center for Galapagos Studies. Again, connected to some of these other groups. I connect to the Museum of Natural Sciences, which is where I used to work, and I'm also an adjunct at NC State University. And so I have a lot of these connections and affiliations because I am really excited about reaching different groups and connecting to different researchers and scientists, and then also still finding a connection through the museum to the public and to students. All these pieces kind of keep me excited about my research so I can get other people excited. And that's what I'm really passionate about is, trying to engage the next generation as well as the current generation in some of this research and making it really relevant for everyone's daily life and understanding.
My biggest takeaway from this episode is how to captivate people. Julie gets excited by being the first to know something and shares it by focusing on why that is new, exciting, relevant, and different than before. What gets people excited about your work? This podcast is hosted by me, Mayla Boguslav, and produced and edited by Joshua Mendel. To learn more about this episode's guest and any cited or named people, take a look at the episode description. Thank you for listening to this episode of the Ignorance Podcast. Please follow the Ignorance Podcast wherever you get your podcasts to learn about science through research questions.
I think the armpits and earwax thing, like I said, really captivates kids and students and gets you thinking. And so that tends to be the fun train wreck to talk about.